Welcome to my presentation. I am Snigdha Bharadwaj, and as you can see on the screen, today we are going to discuss about microencapsulation technique and various method of preparation. In the end, you will be able to know the different types of method of preparation we will use for microsphere or microencapsule particle. So, we will proceed further. First of all, we would like to define the preparation consideration while going to before going to the method of preparation. So, what are the various factors affecting choice of method? So, the method of preparation and its choice are equivocally determined by some formulation and technology related factors as mentioned. As we have discussed in our previous presentation that the core material, the coating material and the other parameters, other ingredients or the method of formulation of microspheres are of important concern. So, the first point represent the importance of the core and the coating material during the process of manufacturing. So, next is your particle size requirement. As I have told you in the earlier presentation that the particle size requirement, which type of particle you want to get through the different type of method of preparations. So, accordingly your method of preparation or the formulation method will change. Drug and protein should not be adversely affected by the process. Yes, of course, if some drugs are thermolabile or thermally affected. So, you will not use the process which is based on the thermal heating, as it may degrade the drug candidate in the process. So, the final or finished product you will get is of no use. Now, reproducibility of release profile and method. This is also of important concern that you are getting the modified release to this kind of method of preparation or not. If the release profile is same as compared to the oral conventional forms, then you have to opt another method of preparation for the manufacturing. Should not lead to stability issues of product. Yes, of course, this is of prime concern that the product you are getting at the end of manufacturing should be stable, as it directly affect the release profile within the body of the drug candidate. Should give non-toxic end product. You are using variety of components in the manufacturing of solid microspheres. So, any of the components should not lead to the toxicity in the system of microcapsules. So, these are the basic primary considerations which first we need to look at and select the selection of the procedure of making microencapsulation is depends on these factors. Now, move on to the next slide. We can have a chart which represent there are variety of procedures mentioned for preparing microcapsules. So, here we can have a look that air suspension method is the method, emulsification, these are the variety of method, then the sub methods are also mentioned here, pan coating, spray drying and spray congealing, phase separation coacervation, solvent evaporation, heat stabilization, multi orifice centrifugation, polymerization, ionic gelation, chemical cross linking. So, these are the variety of procedures we can have during manufacturing of microcapsules, and the subtypes are emulsification may be of single type and double emulsification method, and phase separation coacervation basically it is a process in which we may change some physical condition during the process to get the microcapsules. So, what are those conditions? It is written here, the temperature change, incompatible polymer addition, non-solvent addition and your salt addition, polymer interaction. So, these are the different types of approaches we use in phase coacervation separation technique. Now, if we talk about polymerization, so there may be of three types: normal polymerization, interfacial polymerization, emulsion polymerization. Apart from that, one in situ polymerization is also there. So, we will be discussing these techniques, some of the important techniques out of these in detail. So, first technique is air suspension technique. Okay. So, air suspension technique basically, as you can see in the picture represent the equipment through which we can prepare microspheres or microcapsules using air suspension. Air suspension, as name indicates, the core particle is suspended in the hot air stream onto which the polymer coating is applied through spray. So, in this technique, core material is dispersed in a supporting air stream on which the coating material is sprayed within the coating chamber. Encapsulation efficiency can be varied depending on different parameters like density, surface area, melting point, solubility. So, inlet and outlet operating temperatures also are the factor to be considered. Coating material concentration, what concentration we are using in the manufacturing process, coating material application, cost, overall cost of the material which we are using, and the amount of coating material which we are using in the process. So, as you can see here, the coating chamber is this. Now, as you can see, these are the small particles, and this is the nozzle through which the air distribution plate, control panel, the particle being treated. That means, these are the core particle on which the polymer coating is to be done. Coating chamber, this is the coating chamber, air flow process, and nozzle for applying film coating. The core particles are suspended within the air chamber, and the coating material spray is to be introduced through this nozzle at the high pressure, and it is applied with the help of hot air. It is applied onto the suspended core particles in the air chamber, and it is very efficient technique, So, we can have uniform coating onto the core particles. Now, the second technique is emulsification method. So, as I have told you, two types of emulsification method is there, single and double emulsification. In single emulsification, single type of emulsion has been developed during the manufacturing process, which leads to the preparation of microspheres. So, the aqueous solution and suspension of polymer mixture we use during the manufacturing, and the mixture is stirred and sonicated, and the aqueous solution and suspension of polymer we get after the sonicated in different way, to which heat denaturation, and we can have two types of procedures here. We can apply heat through which denaturation occur of the polymer phase, and the cross linking using glutaraldehyde, formaldehyde, butanol. These are the solvent which are considered to be a cross linking agents. So, microspheres in organic phase developed through heat denaturation, and also in organic phase using cross linking. Both the way we can have microsphere in organic phase, and then the centrifugation, washing and separation microsphere. Basically, you should know what the emulsion is. Emulsion is a system of two immiscible liquid phases which can be suspended or which can be emulsified using emulsifying agent, or miscible using emulsifying agent. Now, in the double emulsion technique, we can have aqueous solution or suspension of drug and polymer, and the dispersion in oil, organic homogenization vigorous. Like if we have two immiscible phases emulsion, then we can have dispersion using some sort of emulsifying agent, we can have the homogenized system, right. So, the two phases will not get separated if we use some sort of emulsifying agent, and vigorous stirring will make the solution or emulsion more homogeneous. So, these are the approaches through which we can have homogeneous system. First emulsion, after applying all these approach, we will get first emulsion. Now, addition to that aqueous solution of PVA, it is a polymer. So, PVA, polyvinyl acetate. So, double and multiple emulsion will be formed, because we have taken the aqueous solution of drug and the organic solvent of polymer, and now we are adding another polymer into the system. That means, we can have double emulsion type of preparation. So, after adding this double second polymer, addition to large aqueous phase, denaturation will occur and hardening will occur due to using this PVA, and we will get microspheres. So, this is how we can get microsphere through emulsification method. Now, polymerization method. So, polymerization can be achieved normal polymerization and bulk, which is categorized into bulk and suspension polymerization. So, now bulk polymerization means we have used monomer single unit, which linked to another monomer with covalent bond and making the polymer chain. So, here this is how we use polymerization process to make microspheres. Now, we will add initiator, so that the cross linking process or the covalent bonding will get started. So, we use initiator, bioactive material with along with monomer, polymer block will get developed, and now after mechanical fragmentation the microsphere we can have. Suspension polymerization means the monomer is here in the system, bioactive material and initiator has been taken in one system mixture, and the stabilizer will be added. The droplet formation will occur after addition of stabilizer, and now after vigorous agitation and heat denaturation you will get the microspheres, and you will separate it out after that. So, this is polymerization, normal polymerization. Now, we will talk about the interfacial polymerization. It involves a reaction of monomers at interface between immiscible liquid. You all know the interface where two immiscible liquids interacts, right. So, liquid phases to form polymer film that essentially developed dispersed phase. So, two monomers we have taken, A in continuous phase and B in also continuous phase. A is dissolved in aqueous phase and B is dissolved in oil phase. Rapid diffusion will occur because oil and aqueous phase are immiscible with each other. So, they will not be mixed with each other. So, you will get this kind of representation here, this kind of system here. So, at the interface, as you can see, small vesicles or microspheres has been formed, particles have been formed. So, you will get microspheres at the interface. So, this is known as interfacial polymerization. Now, the emulsion polymerization, third category of polymerization. Here you will take monomer and aqueous solution of NaOH with initiator and surfactant, like above surfactants we use, right. So, after that adding all these things you will get emulsion, and now this emulsion you will see the polymerization is the simultaneous process which is occurring at the time of emulsion making, as we have added initiator for the polymerization process to be done. And at the end of this process you see the microsphere formation is done and you can separate it out. So, this kind of technique can be used to prepare microspheres. And here the fourth one is phase separation coacervation technique, extensively used technique at the industrial level. So, you can have a variety of microsphere in terms of size, in terms of shape, in terms of morphology and release profiles. So, designed for reservoir type to encapsulate aqueous soluble drugs. Reservoir with complete layering of the polymer, it is not the the matrix. Two types of coating are there, reservoir and the matrix. So, in matrix you will get the meshy layer of the polymer, whereas in reservoir you will get the proper layering of the polymer in the spherical manner. So, designed for reservoir type of encapsulate aqueous soluble drugs, right. Aqueous soluble drugs can be encapsulated using this type of. Now, you see the specifications have been mentioned here, that depending on the type of nature of the drug. So, you can choose your method of preparation accordingly. So, the process basically the principle of this process is of decreasing solubility of polymer in organic phase to affect the formation of polymer rich phase, right. The third component is added apart from the drug phase, aqueous phase and polymer organic phase. The third component is to be added during the manufacturing to support or to enhance the making of microspheres or microencapsules. So, the third component is added to the system, resulting in formation of coacervates. I will show you in respect of pictures, though basically 3 steps are involved in phase coacervation technique. Number 1 is formation of 3 immiscible chemical phases. 3 immiscible chemical phases means drug phase and the polymer phase and the third component phase, right. So, first of all you will make 3 immiscible phases system. Now, deposition of the coating will occur as soon as you add the third phase into the system of polymer and drug. The formation of coacervates or the small droplets will occur soon after adding the third component phase, right. Deposition of coating will occur next to the addition. Now, that means the initiating the microsphere preparation, and then rigidization of coating. Of course, after making small droplets, the strength of microsphere is not so good. So, it takes more time to rigid the polymer coating onto the core material. So, this is the picture through which you can see that these are the equipments or the container which has the propeller or agitator we can see. So, agitation is important to make microsphere. So, agitation is there and these are the jacketed containers in which the polymer and coating and the third component mixture is here. So, liquid, solid, core material storage is here. If you have liquid, then you can have different separate containers. So, through which you can have this polymer solution chamber and the third component chamber. So, here you will mix all together and agitate well. So, using those 5 approaches which I have mentioned in the picture earlier in the same slides. So, you will use different approach like temperature change, like addition of salt. So, these approach will lead to formation of microspheres. By changing temperature, you can have the solidification faster of the polymer coating onto the core material, right. So, drying equipments are attached here and the concentration equipments are attached. How much concentration needs to be added to the main mixture of polymer as well as core drug or the third component. Now, we will talk about the spray drying technique and spray congealing. Basically, these are very well known techniques for preparing microencapsulations. So, here first we talk about spray drying. So, step 1 basically spray drying means the application of polymer solution through a spray onto the core material which are dispersed in the chamber and using heating aid, right. So, that the polymer coating which is in liquid form may solidify onto the core material. So, this techniques involves the core particle are dispersed in a polymer solution and sprayed into a hot chamber, right. So, these solutions are sprayed into a hot chamber or a tank. Step 2, the polymer solidifies onto the core particles as the solvent evaporate. As I have told you that with the aid of heat, the liquid film of polymer when applied to a core material may get solidified when coming into contact with hot air stream, right. The microcapsules obtained are of polynuclear or matrix type, as I have mentioned in the first PowerPoint slides. of the type of microcapsules you may achieve during the manufacturing process. So, it gives the variety, it gives the polynuclear type microsphere preparation. So, you need to consider this fact that if you do not want to use, if you do not want to achieve this multinuclear type, then you cannot get your microspheres done with this spray drying technique. So, it is low cost commercial process and widely used in industry, used for encapsulation of fragrances, oils and flavors. Basically, in perfume industry and in flavor industry and in color industry, it is used widely. Now, the spray congealing means the freezing condition which we provide during the manufacturing process. So, the core material is dispersed in a coating material melt, right. All the things are same in spray drying and spray congealing, only the difference is in the hot and the cold condition environment into the chamber. So, in congealing we use the cold conditions, and whereas in spray drying we use the drying conditions, basically hot temperatures. So, the coating solidification is done by spraying hot mixture into the cool air stream. We use the liquid mixture, hot liquid mixture, but the chamber temperature is cool, right. So, immediate solidification will occur in that case. So, example, vitamins with digestible waxes as you know that the semi solid in nature. So, it is in melt condition or liquid condition at high temperature, whereas it gets solidified at the cool temperature. So, this is why we use waxes to for spray congealing. It solidifies immediately and it will lead to the formation of microsphere, and can be done with spray drying equipment when the protective coating is applied as a melt. So, it can be used with the same apparatus, but the condition of temperature is changed usually. So, this is the equipment. You can see that the air is getting air inlet is here and the heater is here in case of spray drying, and in case of spray congealing the cold air will be application, a cold air application is to be done. Then nozzle will be used for the spraying polymer solution onto the, this is the dry chamber. Cyclone, using cyclone separator we can separate out the suspended particle based on their densities through the system, right. So, here why it is attached, and the rest the air can be removed through aspirator and filtered particles can be separated out. So, this is the assembly of spray dryer and spray congealer. Now, the sixth one is solvent evaporation. It is also a well known technology for preparing microspheres. The active drug and the polymer solution needs to be mixed and it will form the organic polymeric phase. Now, the addition of stabilizers and the other excipients will lead to the formation of oil and water emulsion type, right. And if we increase the temperature, then when we increase the temperature, the polymer which is soluble in the organic phase will get vaporized. So, once it is vaporized, the polymer coating will get solidified onto the core material, that means formation of microsphere. Once the polymer is added in this organic solvent, it is in soluble form. Once we rise the temperature of the system, the organic phase will get vaporized and removed from the system, and resulting in the making of polymer coating onto the drug candidate, right. So, at the end you can see the particle formation of polymer precipitation will occur and the polymeric microparticles will be formed. So, this is how we can achieve the microparticle using solvent evaporation. Now, move on to next slide. It represents band coating. It is a widely used technology for preparing microcapsules and for coating, film coating to apply sugar coating on the normal tablets or conventional dosage forms. So, here you can see the apparatus. two pipes are two pipes are fixed in the tank, cylindrical tank, through which one pipe is for air stream, hot air stream, and another pipe is for spray of polymer coating, and these dot particles represent the drug material which are kept inside the container. Now, the hot air stream will lead to the evaporation of coating or vaporization of solidification of coating, and the another pipe is for spraying polymer solution onto the tank is continuously rotating on its axis, so that uniform coating can be achieved. So, solid drug particles are mixed with the dry coating material in a pan. The rise in temperature causes melting of coating material, resulting in encapsulation of core particle, which further solidifies by cooling. So, this is how we can achieve the coating on the polymer coating on the granules, very effective techniques for coating granules or coloring granules. This is multi-orifice centrifugation technique. It is a mechanical technique, basically it is a mechanical process that utilize centrifugal force. So, I will show you the technology. So, here you can see that the coating material is coating inlet is there and material grooves is there. So, coating membrane. So, when coating material comes through this inlet, it will be in contact in this coating membrane, leading to the formation of thin film. Through this inlet, the core, that means the material to be encapsulated, will be introduced through this slot, and you see the membrane of polymer is on the tray, making tray continuously, and the core material is spraying onto this polymer membrane, leading to the formation of your microsphere. This is very important technique, and all mechanical process which leads to the formation of your microspheres, right. So, coating solution upon leaving the orifice are hardened, resulting the embryonic or micro encapsulation formation. Variables include the rotational speed of the film, so that the thin and thick film can be formed, core and coating material flow rate, concentration and viscosity of the higher the viscosity of the polymer solution will leads to the greater size of the micro encapsules. So, these are the higher the concentration of polymer, thicker the layer will be formed on the core material. So, these are the various factors and these are the various types of preparations which we use to prepare micro encapsules. So, this is basically your the mechanical process and using the centrifugal force. Centrifugal force means this tray is continuously rotating right at high speed, and onto which the core material is sprayed onto the disc, and it leads to the formation of your microspheres. So, this is how we use different type of formulation methods to prepare microspheres, and I hope you understand the basic concepts of all the techniques I have discussed just now, and these are, apart from this, there are various other techniques also, but these are the widely known techniques for preparing micro encapsules. Thank you and stay safe.